This is an introduction into the hematology lab that you have in your lab manual. And as usual, we're going to go over the important terms first. The first term here on the list is Hemocytometer. Hem refers to blood, cyto cells, meter to count. So basically it's a device that allows you to count blood cells. In the old days, this used to be done with counting chambers. Nowadays, people use cell sorting machines that give them then a printout of the pattern or the distribution of the different white blood cells and the count of the red blood cells per microliter of blood. Erythrocytes are red blood cells, usually abbreviated as RBCs, as you can see here. Erythro comes from the Greek red, cyte is cell. Erythropoiesis is the term that we use for the process of forming red blood cells. It all starts out with a stem cell, the hemocytoblast, and the line that results in red blood cells, well, it goes down this pattern of erythroblasts, and then we have a reticulocyte at the end, and then mature red blood cells that don't have a nucleus, by the way. So erythropoiesis, the term that we use for the formation of red blood cells. Erythropoietin is a hormone from the kidneys that stimulates the formation of red blood cells. Erythropoietin is released at a certain rate, and you can increase the release of erythropoietin by getting yourself in a low oxygen environment. So the erythropoietin release can be stimulated by being in high altitude, for example, where you would have less oxygen in the atmosphere, and then your kidneys would think, oh, we need to make more red blood cells to carry the oxygen around, and it will release erythropoietin. Now, erythropoietin is available as a drug, EPO, and unfortunately it has also been abused for athletes that want to enhance their performance. Erythropoietin injections, of course, are not legal. However, altitude camp, exposing yourself, your body, to a low oxygen environment on purpose, that is allowed, and it has basically the same result. So if you want to increase your red blood cells, you can go to altitude, high altitude, and release the erythropoietin naturally from the kidneys, which is of course completely illegal. There's also people that like to sleep in low oxygen tents or have a mask. That's a possibility too. The idea here is that the kidneys will release extra erythropoietin if they measure low oxygen content in the air that you're breathing. Let's take a look at blood cell count abnormalities. The first term here is anemia. Well, literally translated, it means for no and anemia, blood. Obviously, you can't live without any blood, but it basically refers to a condition where you don't have enough hemoglobin. And a lot of time that goes along with also not having enough red blood cells. The first reason that's listed here as a possibility why you might have anemia is hemorrhagic anemia. And hemorrhagic anemia means that you lost a lot of blood. You were bleeding out, or you had an injury and you lost a lot of blood, and so therefore you were hemorrhaging, and therefore you don't have enough blood left. So an anemia due to a hemorrhage. Iron deficiency anemia is a condition where you don't have enough iron, and therefore you cannot make the hemoglobin, because hemoglobin manufacturing requires the presence of iron. The next one is pernicious anemia. That type of anemia is caused by an inability to absorb vitamin B12 from your diet, and your body needs vitamin B12 to make healthy red blood cells. Sickle-cell anemia, that's a genetic problem where the hemoglobin protein is defective due to a genetic mutation, and the defective hemoglobin is not as soluble as the regular healthy hemoglobin. It precipitates out in red blood cells and causes the precipitated hemoglobin to clump up and deform the red blood cells, and then the deformed red blood cells, they get stuck in capillaries, they rupture, and then you have all of the symptoms of sickle cell anemia. And of course, these red blood cells, they just have an ultra high turnover, and therefore the anemia. Macrocytic, microcytic, and normocytic anemia. These are conditions that are describing the size of the red blood cells. So macrocytic means abnormally large red blood cells. Microcytic refers to abnormally small red blood cells, and normocytic, well, they're normal size red blood cells. Microchromic anemia means that the red blood cells don't have as much color as they should have, so they're more pale than normal red blood cells. So normal chromic anemia, the red blood cells, they would have a normal color, versus microchromic, that means they're more pale, they are not as red, they don't have as much color inside. So still, both of them are forms of anemia. That means you wouldn't have enough hemoglobin. But in normal chromic anemia, the red blood cells, at least they look normal. And microchromic anemia, then the red blood cells look very pale. Next up is thalassemia. That is a genetic problem, just like sickle cell anemia. In this case, also the genetic mutation causes abnormal hemoglobin to be made, and thalassemia is usually characterized by fewer red blood cells and hemoglobin that is defective. Moving on to polycythemia. It's kind of the opposite of anemia. And polycythemia, you have too many red blood cells, and your hematocrit, that measures the percent of your blood that's solid, the cells, you have an elevated hematocrit. Leukocytosis means that you have too many white blood cells. Leukos is white in Greek, and so here in leukocytosis you would have too many white blood cells in your blood. And note that you can spell this either with a K, leukocytosis, or with a C. So that's the only difference between these two terms. Leukopenia is the opposite, and again you can spell that with a K or with a C. In leukopenia you don't have enough white blood cells, so very low white blood cell count. And leukemia, that is the direct translation would mean leukos, white, and emia, blood, so white blood. Well, what it means is that you have way too many white blood cells. It's a form of cancer, and it means that these white blood cells are immature, they don't differentiate, they don't take on a role, they don't work for your immune system and do anything good for you. They're just basically taking up space, and it's way too many of them, and they're starting to displace the red blood cells, so your blood starts looking lighter in color, and it's considered a form of cancer, leukemia. Then let's take a look at the white blood cells in general. So there are 5 types of white blood cells that you need to know about. We can stain them, and then by the presence of visible granules after staining, you can differentiate them into granulocytes or agranulocytes. Among the granulocytes, the ones that do have granules, we have the neutrophil, and that one has a funny shaped nucleus, and therefore it's also called polymorphonuclear leukocyte, PMN. And it's probably best if I draw you a picture. So a neutrophil, that's by far the most numerous white blood cell that you have. It's about 2/3 of your white blood cells should be neutrophils, and they do look kind of interesting. They have this, let me just try to draw this. So they have a segmented nucleus. It almost looks like as if somebody punched the nucleus and it shattered into pieces. So we call these lobes. 3 to 5 lobes. I drew 4 right here. So this would be a typical image of a neutrophil after staining. Now it's listed as a granulocyte, but it doesn't have any visible granules after staining, and that is because we don't put it into a neutral dye, and the neutrophil, likes, as the name indicates, it likes a neutral dye. Next up, the eosinophil and basophil. Let me just get that drawn out. Okay, so here are the drawings of these 5 different types of white blood cells. We just finished the neutrophil, so that's in the granulocyte category. The neutrophil is about 2/3 of your circulating white blood cells, and they are characterized by the segmented nucleus, so we call them polymorphonuclear leukocytes. the eosinophil stains with the more acidic kind of dye eosin, and it's a orangey red dye. That's why I drew the cell an orange reddish. The granules pick up that eosin dye, and they show up as orange granules. It's a relatively rare white blood cell. Only about 1% of your white blood cells in circulation are eosinophils. They're really good about sort of eukaryotic parasitic infections. They will try to fight pinworms, for example. Basophils, you have very few of them in circulation because they almost immediately after manufacturing them, they will be migrating into tissues where they're then called mast cells. And they're granules like a basic dye, and therefore you offer them one to stain the granules. So they stain with our crystal violet, which is a deep purple kind of dye. And you can see the nucleus and the granules showing up in very dark purple kind of color. Again, when they are in the tissues, they're called mast cells. They're filled up, these granules are filled up with histamine, and once you release that, of course, you have these more inflammatory kind of allergic type of reactions after that. Moving on to the agranulocytes, we have two of them, the lymphocyte and the monocyte. The lymphocytes are your third line of defense. They differentiate into B cells and T cells. I should write that down. Where the B cells are the ones that are making antibodies, and the T cells are responsible for your cell-mediated immune functions. The monocyte, here our last of these white blood cell types, the monocytes, that's the largest of the white blood cells that you have in circulation. It's about 5% of your circulating white blood cells are monocytes. They have this very characteristic kind of kidney-shaped nucleus, and when they are moving into tissues, then they're called macrophages, where they would be eating up bacteria or cellular debris, whatever needs to be eaten up. So monocytes, once they move into tissues, then they're called macrophages. So they change name depending on whether they are in circulation or they're moving into tissues, just like the basophil becomes a mast cell. Let's move on here to these more terms up here. So acidic versus acidophilic. So the eosinophil is the acidophilic kind of white blood cell. It likes to stain, the granules like to absorb this acidic dye, the eosin, and the basophils, they like this, they're attracted to a basic dye, so we're offering them one, this crystal violet kind of dye. And so in the classic staining protocol where you are staining these white blood cells, you're offering these cells what they like. So the eosinophils, they like eosin, and the basophils, they like the basic dye. There is a lab stir exercise where you learn how to do these blood smears and look at them under the microscope, so you should definitely take a look at that. So when it comes to T cells, when it comes to lymphocytes, that is your third line of defense, and that part of the immune system, those are your specific defenses, and all taken care of by these lymphocytes. You have about 25% of your white blood cells in circulation should be lymphocytes. They differentiate into B cells that make antibodies. So antibody-mediated immunity. It used to be called humoral immunity. Some people still use that term. A lot of people refer to it now as antibody-mediated immunity, and that's mediated via B cells, or lymphocytes that have differentiated into B cells. Now your cell-mediated immunity, that's taken care of by T cells. So those are lymphocytes that have differentiated into T cells, and they engage in very specific type of immune functions. And we'll get more to that in lecture when we do this in the immune system lecture. The plasma protein concentration, that's an important number. Those are generic globulins, usually albumin, for example. They are particles that are too large to leave the capillary as you're moving into a capillary bed in your body. And they're providing an important function because they provide colloid osmotic pressure. That means they are a particle that can't leave and diffuse out of a capillary. So that means at the end of a capillary bed, when you have lost a lot of water and of course nutrients and whatnot, you need to somehow be able to recapture the water that went into the tissues. Otherwise you would end up with edema. And so the plasma proteins, they are very important to provide this colloid osmotic pressure that is then helping you to recapture fluids or tract it back by osmosis. Your plasma protein concentration should be somewhere between 7 to 9% of total plasma. That's quite a bit.